Welcome to the Deep Dive, your essential shortcut to mastering complex material. Today, we are undertaking a critical mission, taking that dense chapter on immunology from microbiology for the healthcare professional. And we're building a clear, strategic map of the body's defense system. If you need to understand survival from a biological perspective, this is definitely your deep dive.
It really is the ultimate biological thriller, isn't it? Historically, immunology is it's key to human progress. I mean, think about the difference between the devastation of things like the Black Death or the 1918 Spanish flu,
Right.
which wiped out tens of millions, compared to our modern ability to manage threats like Ebola or, the SARS virus. The core concept, as the source materials really stress, is this constant game of distinguishing self from non-self.
That distinction really is everything. Okay, so our plan is pretty straightforward. First, we'll identify the core players, that's the antigens and antibodies. Second, we'll scout out the specialized infrastructure, where the defense cells are housed and trained. Third, we map out the whole battle plan, the 3 lines of defense. And finally, we'll look at what happens when the system misidentifies the target or goes wrong.
Right. So to start with the, let's say, language of conflict, the antigen. An antigen is, any agent capable of binding to immune components. But here's the nuance, and it's important. Often, the body doesn't react to the entire agent. It only reacts to a tiny segment that can trigger the response. That specific bit is known as the epitope, or the antigenic determinant.
Okay, and these agents get classified by what they actually do,
Right.
right? Like most pathogens are immunogens because they actively stimulate a full antibody response. Makes sense. But I find the haptens really fascinating. These are, low molecular weight compounds, like certain drugs?
Exactly. They can't stimulate an immune response by themselves. They're too small, basically. They only become immunogenic. They only become a threat if they first bind to a larger, high molecular weight carrier protein inside the body.
Huh. So they need to team up first.
Pretty much. And this whole idea leads us straight to the most fundamental concept, self-recognition. How does the body know what's you? This complex tolerance system is governed by the Major Histocompatibility Complex, or MHC. It's coded by genes on chromosome 6.
MHC. Okay.
Think of the MHC proteins as unique genetic ID badges found on almost every single cell in your body. It lets the immune system recognize and, crucially, tolerate autoantigens, which are your own self-antigens.
So if a cell doesn't show the right ID badge, the body flags it as foreign.
Precisely. And that makes the MHC clinically vital, especially when you think about transplantation.
Right. So comparing graphs, the success rate directly links to how closely the MHC matches. You've got autografts, self-to-self, which are the most successful.
Yep, best case scenario.
Then isografts, identical twins, allografts, same species, different genetics.
Which is the most common type of transplant, like a kidney from a donor.
And finally, xenografts, that's different species, like using a pig heart valve.
Exactly. And we see this ID badge concept, this antigen concept, playing out every day in blood typing, the ABO system. It totally depends on surface antigens on your red blood cells and the antibodies that are already present in your plasma.
Which explains why type O is the universal donor.
It lacks the A and B antigens.
And type AB is the universal recipient because it lacks both A and B antibodies in the plasma.
Correct. It also explains the danger of Rh incompatibility, particularly during pregnancy. That can lead to hemolytic disease of the newborn or erythroblastosis fetalis.
Okay, so that's the threat identification. Now for the countermeasures, antibodies, also called immunoglobulins, or IGs.
Right, the IGs. If
Yeah.
you picture that classic Y-shaped structure, it's made of 2 heavy and 2 light polypeptide chains. The tips of the Y, those are the variable V region. That's the highly specific part that binds to the antigen.
The business end, so to speak.
You could say that. But the base of the Y is arguably just as critical. That's the constant C region, or the FC fragment. This bit doesn't bind the antigen itself. Instead, it kind of dictates what happens next.
How so?
Well, it's the region that binds to other immune cells to trigger destruction, often through opsonization.
Opsonization.
Yeah.
That's like tagging the invader,
Yep.
right? Making it easier for phagocytes to grab and eat.
Exactly. It makes them exponentially easier to ingest, like putting handles on a slippery ball.
Nice analogy.
Yeah.
Okay, so there are 5 distinct classes of these immunoglobulins, and knowing their roles seems key.
Absolutely. The most common one in your circulation is IgG. It's the major workhorse, making up about 80% of total serum antibodies, and you absolutely must remember this. It's the only antibody class that crosses the placenta. That gives crucial passive immunity to the fetus.
Wow, okay. IgG crosses the placenta. Got it. What's next?
Then you have IgA. It usually exists as a dimer, like two Ys joined together. You find it mainly in secretions, tears, saliva, breast milk, and all along your mucous membranes.
So it's providing local protection right at the entry point.
Precisely, guarding the gates.
Okay. Then IgM.
IgM is the giant. It's a pentamer structure, 5 Ys linked together. It's the rapid responder. It's the first class of antibody produced during an initial primary immune response, though it doesn't stick around as long as IgG.
First on the scene, but maybe not for the long haul.
Kind of. And rounding out the list, IgE. It's the least abundant, but often the most dramatic. It mediates allergic reactions and defense against parasites. It binds to mast cells and basophils, telling them to release histamine and other inflammatory stuff.
Ah, so IgE is the culprit behind allergies. And the last one.
IgD, mostly found bound to the surface of B cells. Its main job seems to be involved in activating those B cells.
Okay, that covers the antibodies. Let's pivot to the infrastructure supporting this whole defense system. Where are these players born, trained, and deployed?
Right, the logistics. It all begins in the red bone marrow. That's the primary lymphoid organ. It's responsible for hemopoiesis, the formation of all blood cells.
Hemopoiesis. Got it. So everyone starts in the bone marrow. Where do they go for training?
Well, the T cells, or T lymphocytes, need special schooling. Immature T cells migrate to the thymus gland. That's where they mature. Interestingly, the thymus is most active when you're young, and then it shrinks drastically with age. It atrophies.
And that shrinking thymus is a big part of why immunity declines as we get older, right? That immune senescence.
That's a major factor, yes. Fewer new T cells being trained.
Okay, so bone marrow's birth, thymus is T cell school. What about deployment and surveillance?
The deployment network involves organs like the spleen. It's the body's largest lymphatic organ. It filters blood, and its white pulp area is packed with immune cells ready for action.
And lymph nodes. We hear about those swelling up.
Exactly. They're like crucial filtering stations positioned along the lymphatic vessels. They trap antigens from the lymph fluid, and that's where B and T cells often get activated.
Makes sense. So they swell because there's a fight going on inside.
That's basically it. Lots of cell division and activity. And then, strategically placed at common entry routes, you have lymphatic nodules. Think of them as forward bases. We're talking about structures like the tonsils, and also the vast gut-associated lymphoid tissue.
JK.
Yeah.
That includes Peyer's patches in the small intestine.
Guarding the digestive tract.
Yep. Protecting that huge potential entry point.
Okay, infrastructure covered. Now let's dive into the cells themselves, the leukocytes, or white blood cells.
We can broadly group them. First, the granulocytes, named for the granules you can see in their cytoplasm. Neutrophils are the most abundant type. They're rapid responders, really active phagocytes. They engulf invaders and often die in the process, forming pus.
The frontline infantry.
Pretty much. Then eosinophils. Their numbers go way up during allergic reactions and parasitic infections.
Eosinophils.
These common. They're like the chemical warfare specialists, releasing potent mediators like histamine during inflammation and allergic responses. Okay, those are the granulocytes. What about the agranulocytes?
These include monocytes. They circulate in the blood for a bit, then they move into tissues and mature into the large, long-lived macrophages.
Macrophages, the big eaters.
Exactly. They are phenomenal phagocytes, and crucially, macrophages, along with the dendritic cells, DCs, are the primary antigen-presenting cells, or APCs.
APCs, antigen presenters. So they show the enemy's flag to the specific defenses.
That's a great way to put it. Dendritic cells especially are amazing at this. They have these long branching extensions, kind of like tree branches. They're concentrated at high-risk entry points, skin, mucous membranes. Their job is vital. Ingest the pathogen, break it down, process it, and then display fragments of it, the antigens, on their surface using those MHC molecules we talked about.
And who do they present these antigens to?
To the lymphocytes, specifically the T cells, to kick off the targeted adaptive immune response.
Which brings us to the lymphocytes. B cells for antibody production and T cells for cell-mediated immunity.
Correct, the heavy hitters of the specific defenses.
Okay, players and infrastructure are established. Let's talk battle plan. The 3 lines of host defense. We start with the innate defenses, right? Nonspecific and rapid.
Yep. Innate immunity covers the first and second lines. The first line is all about barriers, physical and chemical. Intact skin and mucous membranes are the main physical shield. A really fantastic mechanism here is the ciliary escalator in your respiratory tract.
Ciliary escalator.
Yeah, tiny hair-like cilia constantly beat upwards, moving mucus and any trapped particles, dust, microbes up towards your throat.
Now you can swallow it.
Exactly. You swallow it, and it gets destroyed by the stomach's incredibly high acidity. We're talking a pH around 2.
Wow, that's harsh.
Very. That acidic environment, plus the slightly acidic pH of your skin, maybe 4.5 to 6, and things like sebum, they form critical chemical defenses.
And didn't the text mention lysozymes? Enzymes in tears and saliva?
Absolutely. Lysozymes are great. They specifically break down bacterial cell walls, found in tears, saliva, sweat.
Okay, so first line is barriers. What if something breaches that barrier?
Then the second line kicks in. This is still nonspecific, but it involves cells and chemicals in a rapid inflammatory response. The key cellular action here is phagocytosis, the cell eating.
Right. And the book broke it down into 4 steps.
It did. First is chemotaxis. The phagocyte moves towards chemical signals released at the site of injury or infection. Second is adhesion. It has to attach to the microbe. Third, ingestion. The phagocyte extends pseudopods, engulfs the microbe, and forms a little vesicle called a phagosome.
And fourth is digestion.
Exactly. The phagosome fuses with a lysosome, which is full of digestive enzymes, forming a phagolysosome. That basically destroys the microbe inside.
Brutal. Okay, so phagocytosis is happening. What about inflammation itself? The redness and swelling?
Inflammation is a protective mechanism, even though it feels uncomfortable. It's characterized by those 4 classic signs you mentioned: rubor, redness; calor, heat; tumor, swelling or edema; and dolor, pain.
What causes all that?
Well, tissue injury triggers the release of chemicals. One is bradykinin, which directly stimulates pain receptors. Ouch. It also stimulates mast cells and basophils to release histamine, and histamine is key here.
What does histamine do?
It causes local blood capillaries to dilate, get wider, and become more permeable, leakier.
Ah, so that's the redness, heat, and swelling. More blood flow and leaky fluid.
Precisely. It floods the area with immune cells, antibodies, clotting factors, everything needed to fight the infection and start repairs.
Makes sense. What else is in the second line? Fever?
Yes, fever or pyrexia. It's a systemic response, body-wide, regulated by the hypothalamus in your brain. It happens in response to pyrogen substances that trigger fever. These can be released by your own white blood cells or directly from microbial toxins.
Is fever always bad? We usually try to lower it.
Well, a very high fever can be dangerous, but a mild to moderate fever is actually protective. It can inhibit the growth of some microbes, it reduces the availability of iron, which microbes need to grow, and it actually stimulates or speeds up some immune reactions.
Interesting. So a low-grade fever might be helping.
It often is. Also in the second-line arsenal are key chemical mediators. We mentioned histamine, but there are others like interferons.
Interferons?
Yeah.
They deal with viruses, right?
Primarily, yes. They're glycoproteins produced by cells already infected with a virus. They don't save the infected cell, but they signal to neighboring uninfected cells. They tell those neighbors to put up their defenses, making it harder for the virus to replicate if it gets in.
So they interfere with viral replication. And they're species-specific, but not virus-specific.
Exactly. Human interferons protect human cells from many different viruses, but they wouldn't protect, say, mouse cells.
Okay. And cytokines, that term comes up a lot.
Cytokines are just the general term for chemical messengers used by immune cells to communicate with each other. They regulate the whole immune response. Think of them like text messages between cells. Interleukins are a major group of cytokines.
Got it. One more big one in the second line. The complement system. Sounds complicated.
It can seem that way. Don't just memorize the 3 activation pathways: classical, alternative, and lectin. Think of it as a biological cascade. It's a system of about 20-30 soluble proteins, normally inactive, floating in your blood plasma. C1 through C9 are the main ones.
Cascade. Like dominoes falling.
Exactly. Once activated by one of those pathways, the classical pathway needs an antigen-antibody complex. For instance, one protein activates the next, which activates the next, in a specific sequence.
And what happens at the end of the domino run?
The end result can be several things. It enhances inflammation, it acts as an opsonin to improve phagocytosis, but the most dramatic outcome is direct cell lysis. The final complement proteins, particularly C9, assemble together right on the surface of the target microbe. They form a structure called the membrane attack complex, or MAC.
Wait, a membrane attack complex? That sounds intense.
It is. It literally punches large circular holes right through the microbe cell membrane. All the contents leak out, and the cell ruptures and dies.
Wow, okay, so complement is a powerful weapon.
Very powerful part of the innate second-line defense.
All right, barriers breached, nonspecific forces engaged. Now we move to the elite forces, the third line of defense. This is the adaptive, specific, and lasting immunity.
Correct. This relies on the lymphocytes, B and T cells, and crucially, it has memory. It's split into two main branches.
Let's start with cell-mediated immunity. That's the T cell's job.
Yes, primarily managed by T cells, but T cells aren't independent operators. They need to be activated first.
By those antigen-presenting cells, the APCs, right?
Exactly. An APC, like a macrophage or dendritic cell, has to present the specific antigen fragment to the right T cell. Once activated, you have different types of T cells. The primary fighters are the cytotoxic T cells, sometimes called killer T cells. They usually have a surface marker called CD8.
And what do they kill?
They patrol the body looking for your own cells that have become infected with viruses or other intracellular pathogens, or even cancerous cells. When they find a target cell displaying the antigen they recognize, they bind to it and release lethal chemicals like perforin and granzymes.
Perforin, like perforate.
Because it punch holes? Yep. Perforin creates pores in the target cell membrane, similar to complement's MAC. Granzymes are enzymes that enter through those pores and trigger apoptosis-programmed cell death. Basically, it tells the cell to self-destruct.
Efficient, so those are the killers. What about the other main T cells?
Ah, the helper T cells. These usually have the CD4 marker. They are arguably the most important cells in the entire adaptive immune response.
Why is that? They don't kill directly.
No, they don't kill directly, but they are the master regulators, the conductors of the orchestra, if you will. Once activated by an APC, helper T cells release a flood of cytokines, those chemical orders we talked about. These cytokines stimulate the cytotoxic T cells to proliferate and start killing effectively. They also are absolutely essential for activating the B cells to produce antibodies. Without helper T cells, the whole adaptive response pretty much grinds to a halt.
Wow. So they're critical commanders. That explains why HIV, which targets helper T cells, is so devastating.
Precisely. It takes out the command structure.
Okay, that covers cell-mediated. What's the other branch of adaptive immunity?
Humoral immunity. This is the domain of B cells and the antibodies they produce. Humoral refers to the body fluids like blood and lymph where antibodies circulate.
So how do B cells get activated?
Similar to T cells, they need to encounter their specific antigen, but the process of activation, called clonal selection, is key here. Each B cell is pre-programmed to recognize just one specific antigen epitope. When that antigen binds to the B cell's surface receptors,
The right key fits the lock.
exactly, that specific B cell gets selected and activated, usually with help from a helper T cell as well. Once activated, it undergoes rapid cell division proliferation. It makes lots of clones of itself. And these clones then differentiate into two main cell types.
Which are?
Short-lived, high-output plasma cells. These are basically antibody factories churning out huge amounts of antibodies specific to that initial antigen. And the second type is long-lasting memory B cells.
Memory cells. That's the key to long-term immunity, isn't it?
Absolutely. That memory is why the adaptive immune response is so powerful. Let's compare the first time you encounter a pathogen versus the second time. The first time, that's the primary response. There's a lag period while clonal selection happens, B cells proliferate, and plasma cells start making antibodies. The main antibody produced initially is IgM, followed later by IgG. It's relatively slow and not super strong.
Okay, so you might get sick the first time.
Often, yes. But now, because you've created those memory B cells and memory T cells too, the second time you encounter that same pathogen, you get a secondary immune response. This response is much faster, much stronger, and lasts longer. Those memory cells are already primed, they get activated quickly, proliferate massively, and pump out huge amounts of antibodies, mostly IgG this time.
Leading to a much higher antibody titer or concentration in the blood.
Exactly. Often, the secondary response is so fast and strong, you eliminate the pathogen before you even feel sick.
And this fundamental difference between primary and secondary responses is the whole basis for vaccination and booster shots,
Yep.
right?
Precisely. Vaccines give you that primary exposure safely, so your body builds memory. Boosters re-stimulate that memory for an even stronger, longer-lasting protection.
Okay, that makes perfect sense.
Yeah.
This leads us naturally into how we actually acquire immunity. The book talked about active versus passive immunity.
Right. It's about how you got the protection. Active immunity means your own body did the work. Your immune system was stimulated to produce its own antibodies and memory cells. This provides long-term, often lifelong, protection.
And you can get this naturally.
By actually getting sick with the disease and recovering, your body fights it off and remembers.
Or artificially.
Through vaccination. You're given a safe, controlled exposure to antigens maybe from a weakened or killed pathogen, just fragments of it, inactivated toxins called toxoids, or even just its genetic material like in mRNA vaccines. Your body mounts a primary response and builds memory without causing the full disease.
Okay, so active is your body making memory. What's passive immunity?
Passive immunity is when you receive antibodies that were made by someone else or another animal. It's like borrowing protection. Because your body didn't make the antibodies or the memory cells, this protection is only temporary. The borrowed antibodies eventually break down.
And this can also be natural or artificial.
Yep. Natural passive immunity is classic mother-to-child transfer. Maternal IgEs cross the placenta during pregnancy, giving the baby protection for the first few months of life. Also, IgA and some IgG are passed through breast milk, especially the early milk called colostrum, providing protection in the baby's gut.
Okay. And artificial passive immunity.
That's when you get an injection of preformed antibodies, often called antiserum or immunoglobulin therapy. This might be used, for example, after exposure to something like rabies or tetanus if you weren't previously vaccinated, to provide immediate temporary protection while your own immune system, hopefully, starts to respond.
Got it. Active is long-term, self-made memory. Passive is short-term, borrowed antibodies.
That's the core difference. Now, finally, we have to acknowledge that this incredibly complex system, well, it isn't always perfect. Sometimes it goes wrong. The text outlines 3 main categories of immune system disorders.
Okay, what's the first category?
Hypersensitivity reactions. This is basically an overreaction, an excessive or harmful immune response to antigens that most people would tolerate. We often call these allergies when the antigen, now called an allergen, is harmless, like pollen or pet dander.
And these are grouped into 4 types, types I through 4.
Correct. Types I, 2, and 3 are all mediated by antibodies. Type I, immediate hypersensitivity, is the classic allergy response. It involves IgE antibodies binding to mast cells and basophils. When you encounter the allergen again, it cross-links the IgE, causing those cells to release massive amounts of histamine and other mediators.
Leading to symptoms like hay fever, hives, asthma, or in severe cases, life-threatening anaphylactic shock.
Exactly. Type 2, cytotoxic hypersensitivity, involves IgG or IgM antibodies binding directly to antigens on the surface of your own cells. This leaves the immune system, often via complement or cytotoxic cells, to destroy those cells. The classic example is an incompatible blood transfusion reaction where antibodies attack the transfused red blood cells.
Okay, and type 3?
Type 3, immune complex hypersensitivity, happens when you have lots of soluble antigens reacting with antibodies, usually IgG, in the bloodstream. They form clumps or immune complexes. These complexes can get deposited in tissues like blood vessel walls, kidneys, joints, and wherever they land, they trigger inflammation and damage via complement activation. Serum sickness is an example.
So types I2 and 3 are antibody-driven. What about type 4?
Type 4, delayed type hypersensitivity, is different because it's mediated by T cells, specifically helper T cells and macrophages, not antibodies. The reaction takes longer to develop, typically 24 to 72 hours after exposure. A classic example is contact dermatitis from poison ivy, or the reaction to a TB skin test. T cells release cytokines that cause inflammation and tissue damage.
Okay, so hypersensitivities are overreactions. What's the second category of disorders?
Autoimmune diseases. This is where the immune system loses its ability to distinguish self from non-self. It loses immunological tolerance. It mistakenly attacks the body's own tissues and cells as if they were foreign invaders.
And there are many examples, right?
Sadly, yes. The text mentions multiple sclerosis, MS, where T cells attack the myelin sheath, insulating nerve axons in the central nervous system. Also, systemic diseases like systemic lupus erythematosus, SLE, where antibodies attack various components of cell nuclei, affecting multiple organs, and rheumatoid arthritis, RA, primarily attacking the joints.
A breakdown in self-recognition. And the third category?
Immune deficiencies. This is the opposite problem. The immune system is too weak or unable to adequately protect the body. These can be primary, meaning they are genetic or developmental defects you're born with. The most severe example is SCID, severe combined immunodeficiency, the boy in the bubble syndrome, where both B and T cell functions are basically absent.
Or they can be secondary.
Meaning they are acquired later in life due to external factors like infections, malnutrition, medical treatments like chemotherapy, or certain diseases. The most well-known example, of course, is AIDS, acquired immunodeficiency syndrome, caused by the HIV virus, which specifically targets and destroys those critical CD4 helper T cells.
Taking out the command center again.
Exactly, leading to a severely weakened immune system unable to fight off opportunistic infections and cancers.
Wow. Okay, that covers the disorders. So to kind of sum up this entire deep dive, the immune system is this incredibly layered defense. You've got physical barriers, then rapid non-specific chemical and cellular forces like phagocytes and complement. And finally, this highly specific, targeted, memory-based response involving T cells and B cells producing antibodies. It's a constant balancing act.
It truly is an immense complexity that ultimately determines survival. It's one of biology's greatest marvels, really. And, speaking of that aging component we touched on, that immune senescence, the decline with age, we noted it's particularly linked to the thymus shrinking, the atrophy of the thymus. Now, given that older adults might still retain a good population of those powerful memory T cells, even if they're producing fewer new helper T cells, how might future therapies thinking way beyond just standard vaccines be designed to specifically leverage or maybe compensate for those existing experienced memory populations?
Boosting the effectiveness of the veterans, maybe.
Perhaps. It's a really interesting question, and it's something that drives a lot of medical research today. How do we best support immunity across the lifespan?
A fascinating challenge indeed, especially for future healthcare professionals listening. Something to think about. Well, with that thought, we'll wrap up our deep dive into the fundamentals of immunology based on this chapter.
A warm thank you from the Last Minute Lecture team.